Replication of the lagging strand is a little bit more complicated, right? So remember that the lagging strand cannot be built as one long piece because the DNA polymerase does not know how to run backwards. So instead, it's constructed as these individual chunks that need to be stitched together. The individual fragments are called Okazaki fragments. They're titled after the scientist who discovered them. And we also have to introduce now one more important enzyme that participates in this process. This enzyme is called DNA ligase. And DNA ligase will come in and stitch together all the individual pieces of the DNA as it's been constructed to make one long polynucleotide strand. So what we're going to do next is we're going to go through the steps of building the lagging strand. And on the next few slides, we're actually going to look at the bottom left-hand quadrant of this particular image. So we're going to look at the lagging strand that's built down here. And when I flip to the next slide, I'm going to point out that the origin of replication is shown on that slide, as is the part of the primer for the leading strand that's running in the opposite direction here. So let's take a look. All right, so here's our origin of replication, and here's that primer for the leading strand that goes off to the right-hand side here. What we need to do now is we need to fill in the lagging strand, which should be going in the opposite direction here. Now, DNA polymerase can't run over to the left-hand side on this bottom template strand because it doesn't run backwards. It only goes forward, right? It doesn't know, the DNA polymerase doesn't know what to do with this 5' end. So the way that we have to construct this lagging strand is that, first of all, we need to ask the primase to build a new RNA primer that's a little bit upstream of the origin here. And so the primase comes in and builds this little primer. Now, once that primer has been built, there's a nice 3' end here for DNA polymerase 3 to come along, latch onto, and it can then fill in this little section between the two primers. Once it fills in that little section, DNA polymerase 3 will then hop off. So now we've got our first Okazaki fragment from here all the way over to here. We're ready to build a new Okazaki fragment a little bit more upstream. So again, the primase is going to come in, it's going to put together a little primer there. DNA polymerase 3 can come in and fill in the space between the two primers there, making Okazaki fragment 2. Now, while the primase and DNA polymerase 3 are working over here, you'll notice there's another enzyme, a yellow-orange one. This other enzyme is called DNA polymerase 1. And in prokaryotes, DNA polymerase 1, its responsibility is to remove all of these RNA nucleotides that were put in there as primers, remove them all because you don't want RNA in your DNA, and instead replace them with DNA nucleotides. So you can see here the DNA polymerase 1 has come in, taken out all the RNA nucleotides and replaced them with nice blue ones, as 2 more to go here, to make sure that you have one continuous DNA strand. And then the last thing that has to come in here to really seal the deal is the DNA ligase. So none of the polymerases can actually seal the backbone, the sugar phosphate backbone between Okazaki fragments or between the leading and the lagging strand. So this is where, and I don't know, it's kind of hard to see, but there's a little bit of a break here in that backbone. DNA ligase is going to come in and it's going to stitch together that backbone so that you have one continuous DNA strand running from the leading strand to the first Okazaki fragment to the second Okazaki fragment, and so on and so forth. All right, so let's take a look at this animation for building of the lagging strand.
The other new strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction. The resulting segments are called Okazaki fragments. Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand. Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding proteins, shown here in blue, and makes an RNA primer to begin an Okazaki fragment. The enzyme DNA polymerase then adds the complementary DNA nucleotides to synthesize the rest of the fragment. The assembly process continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments. After the fragments are made, another kind of DNA polymerase replaces each RNA primer with DNA. Next, the enzyme DNA ligase links the Okazaki fragments to form the lagging strand.
Okay, so now let's put everything together. The leading and the lagging strand are actually built at the same time, and they're built for both the top template strand and the bottom template strand. So what we're going to do is we're going to go back to our replication bubble that we've been working with, and this time we're still not going to look at the whole thing. We're going to look at one complete half, so the left half here. The origin of replication is somewhere off the slide here, but we're going to look at building of the leading strand on the top and building of the lagging strand there on the bottom. So for the leading strand first, the primer is somewhere here underneath my face in the webcam. The DNA polymerase 3 will latch on, and adding new nucleotides to the free 3' end, it will chug along here all the way down behind the green helicase in this image, and it will continue going and going and going until it runs into another replication bubble, or it falls off the end of the chromosome if it were happening to be a eukaryotic chromosome. Now on the bottom template, things are a little different, right? We can't run in the opposite direction if we are a DNA polymerase. So the bottom strand needs to be built in chunks. So you can see there are 1, 2, 3, 4, 5 Okazaki fragments that are being built. I'm going to start with the 5th one. This is the most recent one that has been created, but it kind of starts at the beginning of the process. So the first thing that happens is the primase comes in and it lays down an RNA primer, 5' to 3'. DNA polymerase 3 can then come in, latch onto the primer, and start filling in the gap between primers. And that DNA polymerase 3 here is building the 4th Okazaki fragment. Once the DNA polymerase 3 fills in the gap, then DNA polymerase 1 can go back in and replace all of those RNA nucleotides that form the primers with DNA nucleotides instead. And it can almost finish the whole process. The only thing that it cannot do is it cannot seal the sugar phosphate backbone between Okazaki fragments and also between the leading and the lagging strands. So this is lastly where the DNA ligase is going to come in and seal that sugar phosphate backbone to complete one complete strand, essentially. Okay, and then this slide is a really good summary. It's from your textbooks. It gives you essentially the lowdown on every single protein and enzyme that's involved in bacterial DNA replication. So we talked about the helicase, which unzips the DNA and uncoils it. Single-strand binding proteins, which stabilize that single-stranded DNA. And then you've got the topoisomerases that will break DNA ahead of the replication bubble, unwind it, and stitch it back together carefully so that the DNA doesn't break because of overwinding pressure. We talked about the primase, which is an enzyme that adds that RNA primer so DNA polymerase has a platform to jump off of. And then we also talked about 2 different DNA polymerases in bacterial cells. DNA polymerase 3 adds the majority of new nucleotides when building the new DNA strands. And DNA polymerase 1, its responsibility is simply to replace RNA nucleotides from the primers with DNA nucleotides to make a complete DNA strand. And then the DNA ligase will come in and it will stitch together the sugar phosphate backbones wherever there was a break left behind by this replication process. So either between Okazaki fragments or between the leading and the lagging strands. And then lastly, the way that DNA replication has been presented to you in the animations and in the pictures from your text, it seems like the DNA is stationary and all the proteins and enzymes come in, do their job, and then leave. What we now know is that it's actually a little bit different in real cells. In real cells, it seems like the proteins make a big machine. Make this big complex. So things like the helicase and parts of the DNA polymerases all kind of hang out together as this big DNA replication machine, and they pull the unduplicated DNA in and then push out the duplicated DNA. So it's more like the DNA itself is moving rather than the proteins. And so I really encourage you guys to check out this BioFlix DNA replication animation that I have for you in Canvas. If you can't watch it for whatever reason, let me know and maybe I can send it in a different format. But it's a really good way to get a nice visual of this replication machine.